Here we have the neuron. It receives and processes incoming information, generally taking place in the dendrites and cell body, while the axon conducts electrical impulses and is covered by a myelin sheath, which speeds up the nerve impulse. Now we're going to zoom in on the synaptic region, which is the area between two neurons. In the axon terminal, we have vesicles that contain neurotransmitters. The impulse from the neuron opens calcium channels, which cause the vesicles to fuse with the membrane and release their neurotransmitters, which bind to the receptors on the dendrite of the next neuron. Now, we have the membrane of a neuron. It contains sodium-potassium pumps and sodium-potassium ion channels. The neuron typically has a greater concentration of potassium ions on the inside and sodium ions on the outside. Once the sodium starts entering the cell, we have the depolarization, which is labeled number 2 on the graph, and then more and more sodium channels start opening up, which is the rising phase, labeled number 3 on the graph. This leads to the action potential, which is where the neuron pulses and is the peak of the graph. Then, falling phase, labeled number 4 on the graph, occurs as the sodium channels become inactivated and potassium enters the cell, driving the voltage back down. And at step 5, the sodium-potassium pump regulates the ions of the cell and gets us back to step 1 again. The botulism toxin. It is the light chain of the toxin that depletes the neurotransmitters, and the heavy chain gets released into the synapse, ruining the function of the receptor proteins. The botulism toxin binds to the vesicle's membrane, and the light chain is able to enter it, stopping the release of acetylcholine. The toxin is used in a controlled injection into the face to stop the contraction of muscles, which we call Botox, but it can be lethal in the body.